Thank you so much, Seth. So, as Seth mentioned, I do currently work at the Pacific Northwest National Lab. The work I will be presenting today, though, has been a collaboration with the University of Colorado. I also have a joint appointment there in the Department of Biostatistics and Informatics in the Colorado School of Public Health, which has really been driving this desire and need with my collaborators to start looking at risk factors and omics data in a machine learning framework. And why that is, I will give you a little bit more context a little later on. What I've realized when I've given a lot of presentations is that people don't always know where PNNL is or even much about the Department of Energy National Lab System. So I like to start off just giving that context. So PNNL sits up here in Eastern Washington State. It's one of 17 national labs. The national labs did not exist before the Manhattan Project. The Manhattan Project was really what created the research that set up the infrastructure for the national labs. So those first three were Los Alamos, Oak Ridge, which is in Tennessee, and at that time, Hanford. Hanford is now a cleanup site, and Hanford evolved into the Pacific Northwest National Laboratory. So each of these labs is stewarded by a different mission space. So we are an Office of Science laboratory, where some other labs might be a defense laboratory. And so each of us have different missions and area spaces that we work in. As I mentioned, we are up in Eastern Washington. So unlike what most people often think of Washington State, we live in the desert. So you can see outside of the nice irrigated area, it's all dry. But the campus itself is quite nice. As you can see, it's a very open campus. We don't require gates to get on campus. Everything is monitored through prox access. The other thing I wanted to mention about the lab is we are multi-program. Not all labs are, but our lab is. And so you can see on that little pie chart on the top, we do work for a variety of areas. Basic science, this sort of falls under that one, energy, environment, national security. And then another area related to national security. Nearly half our business is national security related. And we're of moderate size in terms of national labs. We have over 5,000 staff currently. So why PNNL when we're Department of Energy lab in terms of the biomedical space? We do a lot of different research in the biomedical space, but one of the reasons is a core area that we have at the lab is mass spec-based omics. So everybody on here is familiar with MS-based omics, proteomics, metabolomics, and lipidomics. And so in that case, you may have heard of PNNL, as we are one of the largest facilities doing this work across the nation, which gives us a sort of advantage in terms of throughput. We also have a lot of specialized techniques, such as single-cell proteomics. So this is a core area that really facilitates our biomedical research, where we're trying to gain more fundamental mechanistic understanding of the diseases that we're studying. Of course, it's a cross-cutting capability, which is great for us because it needs to meet the mission space of the Department of Energy as well. So why type 1 diabetes? So type 1 diabetes is one of the most common diseases for children. So everybody probably knows a child or an adult who has this type 1 diabetes. It is growing, as you can see here in this slide from the TEDdy study, which I will talk about a little bit later. But the challenge with diabetes, which I'll discuss more in the next slide, is that there is no cure. There is a treatment. That's insulin, but that means you are on insulin the rest of your life, and all the comorbidities that go along with having this long-term chronic health decision of type 1 diabetes. So as you can see from this figure, which I took from the TrialNet study, type 1 diabetes progresses in stages. It's an autoimmune disease, which means there is some trigger event that leads to an immune activation. In type 1 diabetes, this means the beta cells in the pancreas are attacked. And and the immune response of it eventually kills them off. So as we know with autoimmune diseases, it's usually a specific organ or cell type that is attacked by the person's own immune system. It's the beta cells that are necessary for normal insulin production, which is why people with type 1 diabetes have to take insulin. When and how long this immune response starts and progresses is not well understood. However, research subjects usually categorized into 4 stages as seen here. The first stage is when you have normal blood sugar. This is the stage 1. But you have the start of type 1 diabetes, which means you can measure the autoantibodies in the child or adult. There's adult onset, it's just not as common. Stage 2 is when you still are not symptomatic, meaning you don't need insulin, but when you are tested, you have both these antibodies and also abnormal blood sugar. And then finally, stage 3 is clinical diagnosis. And then a lot of people are categorized then finally as stage 4 after a certain number of years as long-standing type 1 diabetics. So the real challenge here is that children go through these early stages at different time courses. Some kids progress by the age of 2, some don't progress till they're 13, 14, or even young adults. So if you're sitting down here in stage 1, 2, pre-symptomatic, you're not part of one of these studies, so you're not having your autoantibodies or blood sugar tested, you won't know that you have type 1 diabetes until you hit stage 3. And so many of these children are diagnosed in the emergency room where a lot of damage has already been done. So the research is really shifting to the left on this figure, trying to understand what's going on at stage 1 and 2 and predict which children are going to hit what stages at what age so that interventions can be developed to try to slow down the progression of this disease. So type 1 diabetes has been studied for decades. It's pretty much a standard mantra when it comes to T1T that 2 children can have identical what we would consider genetic risk, but they progress completely different. As I mentioned, one may progress very young. They may both get autoantibodies at the same time, and then one progresses type 1 diabetes really quickly, and the other one takes a long time. So it's definitely some combination of environmental and innate factors in combination with the genetics that are sort of driving this progression of the disease. So on this slide, you can see people have been doing these birth cohort studies because the only way to know when a child is in stage 1 or stage 2 is to follow them from birth and measure those autoantibodies and other measurements of progression of disease. Each of these studies have had different focus. Some have been more focused on diet. Others have been more focused on genetics. And so today I'm going to talk about 2 of these, DAISY, the diabetes autoimmunity study in the young, and TEDDY, the environmental determinants of diabetes in the young. It's the 2 that I work with the most, but I wanted you to know that there are a lot of studies out there studying different facets of this disease. And as you can see, following thousands of children over time, looking at the blue numbers there on your right. Okay, so I mentioned I want to take classical risk factors, such as genetics, and integrate with omics data to try to make predictions about these stages of disease. So I don't want to give any false impressions. I often get asked about the clinical utility of our work, and although that is where we want to go, we work with physician scientists on this. I don't want to give any false impressions. We are way down in this discovery phase. So this is a paper from a tutorial that we put together on best practices for MS-based biomarker discovery. But you have to really do this in phases. So the whole shotgun proteomics is way down here in the identifying biomarker candidates. Then you need to go through a verification and then finally a real true clinical validation. So we are way up here trying to move towards the dash circle. Okay, so just to restate the goal of the work I'm going to show you. So statistics are wonderful. I'm a statistician by training. However, they give you population-based associations, which can be highly valuable in studying disease. But our goal is to explicitly model the data using machine learning to see if those markers that we are looking at as they are measured, which means they may not validate, this is what's measured in our global data and sometimes targeted data, to predict a future state of the child. So we're trying to find a multivariate panel that targeted assays could be used that would have lower variability and move into verification. So there's lots of challenges, though, when we start looking at different data types, because we don't want to just look at the omics data alone, because we know that it could be the results of that you may need the genetics data and the other metadata associated with that to really get a better understanding of the prediction and the interactions. So some of the challenges, right, is that these are different feature types. If you're looking at SNPs, for example, they're binary. If you're looking at metabolomics, it's quantitative. Proteomics is quantitative, but relative at that. So there's a lot of embedded feature selections for different machine learning algorithms, but the question is, do you really want to throw this all into one single machine learning algorithm? And then finally, we want to make sure that we're reporting all of these features from these different data types in a way that can be prioritized for follow-up. So before I get into the design and the data, I want to make sure I go over terms and what I mean by them. So I think the training and test set are obvious to most people, and that's the portion of the data set used to fit the model, and the test set is, of course, used to evaluate it. So in cross-validation, you parse it up equally. In this context, that is what I mean. Just standard K-fold cross-validation, you'll have training and test sets within that, that parsing of the data. By model selection, I just mean a process for selecting a machine learning model from a set of candidate models. You'll see I don't do anything fancy here, just optimization, but that's what I mean by model selection. And then feature selection is just a process to select features or variables from a data set, and there are lots of ways to do that. And then on the bottom, I put validation set because, as you'll see in the first example I'm going to get, we don't have a validation set. The data is too small. A validation set should be a set that's set aside all the way from the beginning, right? You don't touch it from the beginning in any of the tuning of your model. When your sample sizes are small, though, that can be very difficult. But this is extremely important to have a validation set. Ideally, from even a different cohort, you'll see that we can't do that, at least not yet, because our cohorts don't collect data in the same way, but we at least can create a validation set for the second example. And I'll show you how the results change with that validation set. So here's the study design that we had. This is specific examples for the DAISY cohort, which was set up by our collaborators into 3 groups: controls, those who are autoantibody positive, and those who have type 1 diabetes. Each of our subjects, each of our kids has 4 time points. So you can see this follows the stages of type 1 diabetes. Everybody's negative. You first show autoantibodies. Then you show autoantibodies, but you're not clinically diagnosed yet. This is the last visit before that diagnosis, and then the first visit after diagnosis. So our collaborators asked us to look at 2 specific comparisons. The first is the change here, right? What is changing for those kids, predicting those that are going to progress to autoantibody positive from those who maintain as controls. The second was to look at the change here. So by change, there only are temporal features such as proteomics, metabolomics have changes at each of these time points. But again, what's changing, causing this transition from autoantibody positive to type 1 diabetes? And as I mentioned, this is very interesting, and it's understanding the mechanisms of what is driving the change, but it's still not quite that prediction that I laid out in the beginning, which I'll touch on in the second discussion in regards to TEDdy. So everything I'm going to show you today is in this paper. So if you're interested in learning more, feel free to go read it here. So the data, we had what we considered 6 unique data streams associated with these 70 kids. We had patient metadata. So this is things like age, race, sex. We had GCMS metabolomics data, immunochip data, proteins, and these were actually measured by SomaLogic. We had targeted proteomics using multiple reaction monitoring, and this was the data generated by our lab. And finally, they had cytokine data that had been also measured. So the genetics and the patient metadata are static data sets. That means they are going to be the same regardless of time except for age. And the other 4 data sets are all dynamic, and so they change over time. And you'll see these really drive a difference when we find which markers are important for these 2 different hypotheses. They're quite different. So our approach here was an integrative machine learning framework that embeds the feature selection using a standard optimization method. So we can break this into 4 steps. The first is to find the best machine learning algorithm for each data type. The second is to fuse. You have to, if you're not going to use this one single algorithm for all of our 6 data types, we have to somehow do a fusion. And then do a feature selection, where our goal is to do the feature selection on the integrated result, not on the individual data sets. And finally, after you do this, you have to do a final evaluation of your model and the feature interpretation. Okay, so a little bit more on the fusion. As you can see from this table, right, there are a lot of different benefits and limitations to different approaches that need to be addressed for each data type. Some of our data type are categorical, so some of these methods won't work. There's also constraints that are associated with the distribution of the data. So there's not really a great kitchen sink approach just to throw these all together into one single machine learning algorithm. Although it has been done with random forest somewhat successfully quite a few times. So the only constraint that we put on what machine learning method we can use for any given data set is that it must yield a posterior probability. So that's a probability of observation K, so that's that the child progressed to type 1 diabetes or did not, given that observed data. And then we just do a simple probability-based fusion. If you look in the literature, there's a ton of ways to do this fusion. You can do weighted sums, weighted probabilities, a lot of different approaches. Here we just keep it simple, and we're just doing this simple probability-based. This is one minor weakness in that means we allow each of the data types to have an equal contribution to the machine learning algorithm. So for the feature selection, so I really like the approach taken by random forest. Evaluating after a feature is included in a tree as its importance. We actually have taken the same idea, but use optimization to find a feature solution. So here I'm showing simulated annealing as our strategy and our optimization as the area under receiver operating characteristic curve. But you could use any optimization approach you wanted, and any optimization. This could be F-score or whatever else that you're interested in. But for the simulated annealing, basically, we change one of our features. If it's in the model, we take it out. If it's not in the model, we add it, compute that AUC using cross-validation, and then we compute the simulated annealing probability, where we take that change in the feature based on this probability. And so that also means it's not a purely greedy algorithm. It allows for some variation and some features to be kept even if they might not be 100% increase the AUC. Then we do this process until we converge. So there are a couple of parameters you have to set. You have to set this V, which basically, if you set it to be really high, it means I will only take a AUC value that increases, or you can set it low and allow it to take more features even if they don't increase the AUC. You can do this until it converges, until your AUC changes by a delta. So it's the second parameter you have to set. And then you just repeat this multiple times and you'll get an ensemble solution. So you'll have a set of features for one run, set of features for run 2. And I'll show you what that looks like here in a minute. This capability is available in our Pepper package on GitHub, so feel free to go check it out. Okay, so I covered some approaches, and I just want to remind you of the study design and introduce these terms, seroconversion. So we're going to say seroconversion is the change in these 2 time points, seroconversion to those autoantibodies, and progression is these 2. Progression to type 1 diabetes. All right, so going through that previous sort of steps that I had mentioned, the first is this algorithm selection or model selection for each of our 6 different data types. So this is just showing sort of a colored heat map of if you run each of these data sets alone, using each of these different methods, using cross-validation, no feature selection, which is why the AUCs are so poor. What are sort of the best methods for each of these data sets? You'll see once in a while we don't pick the best method. We pick the one that's most consistent between our 2 different comparisons. In the end, we come down to a random forest for our core omics data, a naive base for our patient metadata and our genetics, and a support vector machine for the cytokines. So the results of the feature selection for each run is a set of features selected based on that optimization run. So as you can see here, at least for this case in the metabolomics data, there's a lot of variability in which features are selected. So I'm not going to talk about this today, but we've looked at thresholds in many scenarios, and 50% seems to give a good result consistently. And we are working on some new methods papers to sort of show this and even do sort of a de facto random model to find out what really is the optimal, but 50% works well. And then you can see which features are selected. The yellow represents the seroconversion endpoint and orange is progression. Consistently, there are no cytokines that make the threshold for every endpoint. And the metadata is a percentage that is maintained the highest, followed by genetics and so forth. So the obligatory ROC curve, which I don't have in the second part, the results here, you can see them in the ROC curve. So the green is our method. The black is if you don't do any feature selection, so not good. I will tell you if you use glucose alone in the progression, which we all know glucose is a marker of type 1 diabetes, you do almost as well as using all of that data. We also ran a recursive feature elimination. And to try to make it sort of apples to apples, we ran the recursive feature elimination many times. So, you know, if you, based on the cross-validation, you'll get different results if you repeat recursive feature elimination over and over. So we did that same thing, and we got an ensemble solution for the features from recursive feature elimination. And you can see the results here as well. So again, dependent upon that threshold. So the features. So there are some in common between our 2 different classifications that we're doing and some that are different. When it comes to the demographics, the age and having a first degree relative, this is not false discovery rate, this is first degree relative. And the FDR here is what we mean, are almost, are selected all the time for both. What's interesting is for the seroconversion, we get additional HLA genetic marker as well as Hispanic. We only have a couple of SNPs in common up above, meaning there may be genetic risk of just progression to autoantibody might be different than progression to T1D. So I didn't draw out all those the names of all of the other omics data, but here is how I sort of present it to my collaborators. That's worked really well. So I know I've been talking about seroconversion and then progression, but I'm going to start here with progression because it is easier to sort of show the obvious connections to what we expect, right? So we can see right at the top, 100% of the time, glucose is always taken. 99% of the time, mannose is taken. These are all sugars. This is not surprising. But this is the reason also when we talk about the TEDdy study that we're doing things differently, because as I mentioned, we're looking at that change from type 1 to type 2, or from stage 3 to stage 4. And since we have measurements of stage 4, it's not really predicting who will be in stage 4. It's telling you which markers. If you had both measurements, would you get that? But if you want to predict from stage 3 to stage 4, you wouldn't use stage 4 data, which we do here. But you also find a lot of proteins associated with diabetes, like this diabetes vascular calcification. But these do give our clinical collaborators direction to go to try to define new hypotheses, as well as various steps. This is the same table, just showing that, when you start plugging these into PubMed or however you want to search, you start to find relevant publications to our top candidates. So seroconversion, here's the same table, but for seroconversion. On the top is ascorbate, which is otherwise known as vitamin C. This has been a long-standing dietary marker of interest being studied. In terms of SNPs, there's also a defined autoimmunity gene, as well as proteins that promote beta cell proliferation. So we can see early markers of beta cell damage happening here. But also other markers where you could start thinking about vitamin C and how that could be used as a protective factor. Again, just as before, ascorbic acid and other related markers have been seen before. Okay, so it looks like I'm doing okay on time. The second discussion on TEDdy is much shorter. So I think, we've done this, what I call ROFI, this repeated optimization for feature interpretation algorithm in a lot of different scenarios. I've shown you one of them here. It seems to work very well in most cases. And our researchers really like that we can use it with any machine learning algorithm and identify these candidate markers that work in combination with genetics and clinical risk factors and present it in this nice either tabular or visual form. So the next steps, as I mentioned, we want to predict one time point from another time point, not a group from another group based on a change between time points, which I will show in the TEDdy study. And we also want to evaluate the approach on a data large enough for validation. And again, this will be what I'll show with TEDdy. These last two are kept in gray, things that we are working towards. Is clinical validation with our collaborators at the Barbara Davis Center for Diabetes, as well as capturing interactions between features. I will admit, I know there's a lot of literature in this area of capturing interactions between features, but if you have approaches you think would fit well with what we're doing, please send them my way. I'll have my email at the end. I would love to think more about this topic. I just haven't a lot yet. Okay, so after this DAISY study, we began collaborating with the TEDdy study with our Colorado PI, Dr. Ruers, who is a co-lead also on the TEDdy study. So TEDdy is a lot larger than DAISY, and they also collect samples every 3 months from birth. So they collect a lot of data. They capture the age of seroconversion to autoantibodies, similar to DAISY, as well as the point at which the kids are diagnosed with type 1 diabetes. So at most, of course, you have kids that miss appointments from time to time, but this means at most you have a 3-month window, or in most cases, a 3-month window to the time point prior to diagnosis. So on the bottom, I tried to just create a little schematic. It's not the best, but you know, what you'll have is kids will seroconvert at various times. That's the open circles. And then you can go backwards in time to the data before the seroconversion. So my collaborator at the Barbara Davis Center is most interested in this very early indicator of disease, this conversion to autoantibodies, but completely pre-symptomatic, so no symptoms. You may ask, why didn't we start with type 1 diabetes, which would have been easier and we probably would have got better results? It's because this was what interested him. We are working right now on predicting which children using infant-based data will progress to type 1 diabetes by age 6. But this is why we focus on seroconversion. He's really interested in trying to find markers that will help him in the clinic find kids early who are going to seroconvert. Again, we had a bunch of risk factors. We had metabolomics, lipidomics, and SNPs. Unfortunately, in this case, the proteomics data has now been generated and is with the Data Coordinated Center for TEDdy, but we didn't have access to it yet at this time, so it's not included. So we did set this up with a validation set. As I mentioned, we didn't have the right overlap of data to create a validation set from one of the other cohorts that have been studied over the years, but we did have 157 cases and controls that were designed by the TEDdy study. So we took 25% of these and set them aside as our validation set. So we kept it separate from any initial filtering that may be done based on statistics, as well as the model selection, and the optimization and the feature selection. So that set was set aside at the beginning and only evaluated once we were done with the whole process that I showed you with DAISY. So again, this paper is published in the Journal of Diabetes. So if you want more details, I'm not going to give methods here because I gave methods previously. There's not very much that we did different from the prior one here, except for it was a very different classification, as I mentioned. And and it was, we had the validation set. So I'm not going to show a ROC curve this time. We do, you know, cross-validation a whole bunch of times, so we can get, when we're doing the model development. So we actually have a lot of AUCs. We typically just show one, sort of the average, but here I thought I'd show that distribution. So we get similar to DAISY in terms of when we use all features for trying to predict seroconversion. The only thing I'll remind you here is that we're not using that time point at seroconversion as we were with DAISY. Our top 42 features, these are our ROFI 50%, are up here. And if we go 0 months, 3 months, or 6 months prior to seroconversion, we get almost identical results. These AUCs are not highly impressive, but given these kids are pre-symptomatic, our collaborators were impressed we could do as well as this. But Unfortunately, when you look at the validation set, still much better than random, 0.63, 0.65, that sort of range. The validation set can obviously be only analyzed once, so we just have one dot. And you see that the performance does drop dramatically with the validation set. So there's a lot more work to be done here, but we still were able to pull out some interesting features to talk about with our collaborators, as well as do some other sort of cross-study comparisons. So here, these are our risk factors and our SNPs. DR34 is a well-known HLA. Gestational age came out, DR33. These SNPs that are underlined, I will tell you why they're underlined, I think, on the next slide. But, here's our threshold, and you see that there's a lot of SNPs, but there's also a lot of lipids and metabolites. For the SNPs, 75% had a putative target gene that had already been related to the immune system. The metabolites and lipids were grouped into 3 pathways: lipid oxidation, phospholipase signaling, and pentose phosphase. Okay, so these are just some plots showing gestational age. As the older you are, the more likely you are to have this progression, as well as this DR34 HLA is the same. The prebiotic exposure and the DR33 are the opposite directions. And then for the SNPs, we actually did have some overlap with DAISY. So there were 4 SNPs that had been measured both for TEDdy and for DAISY. So here we're showing the percentage of children for each of the genotypes. And you can see that the TEDdy on the y-axis and the DAISY on the x-axis correlate really well, actually, like over 0.85. So, it gives us a lot of confidence that, maybe a global genetic risk score might not be the best, but focusing on some very specific SNPs that seem to validate across the 2 studies in terms of prediction. So lastly, as I mentioned, we are squarely in the discovery phase, even with this machine learning. And so we are always looking to put these markers that we find in the context of specific pathways, because understanding the mechanism is actually how it affects the downstream, measurement of these specific things like adipic acid can actually help you in identifying what sort of targets or drugs you might want to start hypothesizing about or evaluating. All right, well, I appreciate your attention today. I will end with highlighting the great team that's collaborated on this work. We've had multiple statisticians working on various aspects, including Lisa Bramer, Sarah Akers, and Brian Stanfield. From the omics side, we receive a lot of guidance and evaluation results by Tom Metz and Ernesto Nakawaisu, and the team at the Barbara Davis Center. They lined up all the evaluations, assembled the data sets, and are helping with further interpretation. And with that, this is my contact information, and I'll give you 1 second in case you want to write it down. It should only take that long. It's bj@pnnl.gov. Quite short, and then I will stop sharing, and I'm happy to take questions.
Thank you very much, Bobbi-Jo. That was really interesting. So we do already have some questions in the Q&A box. Our first is the deep interpretation of the relevant features and interesting to gain biomedical insights. Did some of this match or contradict other literature?
Well, as you saw with when we were going through the tables from the first study, they did match the literature. We didn't have any that actually contradicted, which was good. We had some where we couldn't find any literature for sure, and so we're looking more into those and trying to decide if we want to follow up on them specifically. not this work, but we've had other studies where we've done similar approaches. That we have a grant right now with the Helmsley Trust focused on complement proteins specifically, which came out of an analysis like this, which have been associated with the immune system, but not really studied deeply. But our analysis sort of indicated that might be worthwhile.
All right. So our second question, excuse me, do you think the number of selected features per information level, that is metadata, genomics, proteomics, etc., do you think it reflects on which level the disease has the most impact, or rather the data quality or some other factors?
Yeah, that's a good question. I think it really just comes down to both data quality plus signal, right? So the interesting thing here, I'll share my slide. I'll share this again so I can show you what I mean. There we go. So here, I'm not going to go in the presenter mode, or I can't see. You'll see the cytokines, which are actually, have very low variability, were never picked. They just weren't the right cytokines. This was GCMS metabolomics, which is quite variable, and a relatively larger percentage. So I think it's really about a combination of the two. It's how much variability is in that data, as well as signal. I mean, similar to statistics in a sense.
So follow up from me. You said they weren't the right kind of cytokines. Do you have any indication what would make the right cytokines? Is there any specific features that you could try to focus in on?
So those cytokines were selected based on a prior study. As I mentioned, for example, we're now focusing on complement proteins. So those might be a more informative cytokine to capture. But
okay.
Yeah, it's just for the specific comparisons that we were doing, they weren't well associated or able to predict our endpoints.